Afternoon, one and all gathered here. Today, we are pleased to welcome a very special guest, Dr. Amrita Khaire, who is ICMR Research Associate 2 in the Department of Mother and Child Health, Interactive Research School for Health Affairs, Bharati Vidyapith deemed to be University, Pune. She has been working in the field of maternal nutrition and endocrinology for the last 10 years. She has also a good relevant experience both in human and animal research and has over 25 international publications to her credit. She has received various fellowships from national agencies like ICMR, CSIR. She has also several prestigious awards to her credit, such as Young Scientist Award at 46th National Conference of Nutrition Society of India, then Travel Grant Award by Korean Society for the Study of Obesity, Best Oral Presentation prizes at various national and international conferences. Dr. Khaire was also invited as a president for keynote address at International Conference on Advanced Research in Life Sciences held at Sadhguru Garge Maharaja College, Kharad. She is a research committee member of Jan Seva Foundation, Pune. And today she will share her expert opinion on role of maternal nutrients on cardiometabolic outcome. Now I am requesting our HOD to please welcome Dr. Amrita Khaire.
Thank you so much, Animita ma'am. So, good afternoon, everyone, and good afternoon, Dr. Amrita ma'am. So, once again, on behalf of the Department of Nutrition and Dietetics, I warmly welcome you, ma'am. So, we are so much, will be advantaged by having your lecture. So as many as our students can get benefit from this lecture. So once again, I welcome you, ma'am.
Thank you so much.
Yes, ma'am. So, ma'am, Anita ma'am,
yes, Amrita madam, please you can start. Yes, you are audible. You are audible.
Okay, thank you so much, Anita, and thank you so much, everyone, for inviting me today for this webinar. Hello, everyone, and here we'll be discussing today on the role of maternal nutrients on cardiometabolic outcomes. So, as we all know, there is a high prevalence of non-communicable diseases like obesity, cardiovascular diseases, diabetes, and deaths due to these NCDs are known to account for about 80% of the disease burden globally. And nearly 61% of the deaths in India are attributed to NCDs, out of which around 26% are due to cardiovascular diseases. And this prevalence is known to increase by 15% by the year 2030. They have major economic implications, like NCDs create a significant strain on household budgets, particularly in low-income countries. And these diseases are known to cause billions of dollars in losses of national income each year. And economic analysis in India, it suggests that each 10% rise in NCDs is associated with about 0.5% lower rate of annual economic growth. So this is metabolic disease burden. Metabolic syndrome is a cluster of factors that increase the risk for developing diabetes and cardiovascular diseases. And the major risk factors associated with these metabolic diseases are increased obesity, that is high BMI, adverse lipid profile in the form of triglycerides and cholesterol, elevated blood pressure, hyperglycemia, and insulin resistance. And the prevalence of these diseases, high prevalence, is mainly attributed to the double burden of under and overnutrition and to the nutrition transition. That is a transition from healthy diet to a diet containing high amount of unhealthy fats, such as lacks essential micronutrients, vitamins, antioxidants, and minerals. And consumption of such an imbalanced diet is associated with metabolic disturbances in the body, which subsequently increase the risk of cardiometabolic diseases. So we can say that nutrition plays a very important role in the development and progression of metabolic diseases. And current research, it also suggests that about 50% of these diseases are caused due to imbalances in the maternal nutrition or adverse early life programming. So let's see why it is so. Basically, pregnancy is considered as a vulnerable period for the health and well-being of both the mother as well as fetus. And maternal nutrients, they act as essential building blocks for the overall health, brain development, and for the immune system of the baby. So the nutrition before as well as during pregnancy, it plays very important role in the uteroplacental development, fetal growth and development, and it also influences the risk for developing risk for developing chronic diseases in the adulthood. The first 1,000 days of life, that is during pregnancy as well as during postnatal period like breastfeeding, weaning, and toddler phase, is considered as a rapid phase or a period of rapid growth and development. Any disturbances or alterations during this period is known to increase the risk for non-communicable diseases. So maternal nutrition, poor maternal nutrition, it can be linked to poor birth outcome and development of diseases in later life of the offspring. This concept is basically called as developmental origins of health and disease, and it was first proposed by Professor David Barker in 1980, wherein he showed the association between low birth weight and the risk for developing NCDs in the adulthood. And he postulated that adverse influences early in the development and particularly during intrauterine life can result in permanent changes in physiology and metabolism, which result in increased disease risk in the adulthood. The most elaborately reported human study based on Dohar concept, it is Dutch hunger winter study, which was carried out in which occurred in 1976 in Netherlands. And this study, it demonstrated the association between occurrence of coronary heart diseases in the middle age with the premature birth or low birth weight. This study also showed that the timing of exposure or adverse nutrition during the famine period is a major determinant in determining phenotypic outcomes. And individuals who were exposed to famine as an embryo or fetus, they had a high prevalence of CHD, they had high BMI and glucose intolerance. So maternal nutrition is the primary environmental factor which influences the fetal development as well as postnatal disease risk. This concept is also well demonstrated by using various animal models. As we can see here, the research has been carried out in animals by using global undernutrition model, then macronutrient restriction like protein restriction, carbohydrate restriction, and overnutrition studies such as intake of high calorie diet, high fat diet, micronutrient deficient diets. So all these studies, they showed that any imbalances in the maternal nutrition are known to affect or pregnancy outcomes and also have adverse consequences in the offspring in the form of organ remodeling. Then it also leads to elevated blood pressure and sometimes cognitive deficits. So these are the mechanisms. Maternal malnutrition, it basically leads to maternal and placental abnormalities leading to fetal malnutrition, which subsequently affects the organ development leading to organ malformation, mostly in the pancreatic beta cells, then muscles, heart cells, and also changes the endocrine settings by influencing the levels of hormones and growth factors and causing epigenetic changes, which increases the risk for developing these diseases. So original insult in the intrauterine life as a consequence of maternal malnutrition is known to cause permanent programming of metabolic functions, and these changes are further perpetuated to the next generation. It is now very well demonstrated that the effects of programming are not limited to the first generation, but are also transferred to the subsequent generations through non-genomic interactions. So if we consider this as a F0 generation mother, if we consider this as a F0 generation mother who carries F1 generation fetus in her womb. So if there is any insult or exposure of this pregnant female to an insult, it leads to programming changes in the F1 generation offspring. And this is the F1 generation offspring who already carries the germ cells which will form the F2 generation. So if there are any alterations or changes in the F0 generation, it is known to hamper the F1 as well as F2 generation development. So these changes or mechanisms basically include the changes at the molecular level, like transcriptional regulation, translation changes, and also the epigenetic adaptations. So researchers are trying to find out the mechanisms or mechanistic processes that link early life insults with the adult diseases. This is the epigenetics. It is a major phenomenon. As we know, genetic material is mainly transferred in the form of DNA. But in case of epigenetic changes, it is the study of inherited changes in gene expression caused by the mechanisms other than DNA sequence. So there will not be any changes in case of in the DNA sequence itself, but it will still lead to phenotypic changes in the phenotypic outcomes. And nutrition is one of the major factors influencing epigenetic changes. Epigenetics is the heritable and reversible modifications of DNA and DNA binding proteins, resulting in changes in chromatin structure and gene expression. And these methylation patterns are usually established during embryogenesis and remain largely unchanged during adult life. It basically involves three major processes like methylation, histone modification, and microRNA interference. As we know, DNA is a double-stranded helix which contains the DNA base pairs like ATGC. In case of DNA methylation, methyl groups get added to certain DNA base pairs which repress the activity of that particular gene. And in case of histone modifications, a combination of different molecules get attached to the tails of histone proteins, which alter the activity of DNA wrapped around them and inhibit the gene expression. Now, in case of non-coding RNA interference, we know central dogma, that is double-stranded DNA is first converted to mRNA, and this mRNA is further utilized for the protein synthesis. Now, in this case, there are some small and long non-coding RNA molecules which get bind to the target mRNA molecule and inhibit the gene expression or translation by degrading the targeted mRNA molecule. So scientists are now finding the link between what is the link between diet and epigenetics. Among various nutrients, micronutrients are considered as a major methyl group donors. If we consider this is the food that we ingest, which contain different types of micronutrients like folic acid, vitamin B12, dietary methionine, choline, which act as a source of methyl groups, and these methyl groups further carry out the epigenetic reactions. So these nutrients modify the epigenetic phenomena and alter the expression of genes at the transcriptional level. So as we know, the functions of maternal micronutrients, they play important role to meet the physiological changes in mother as well as for the fetal growth and development. They mediate cell growth and various signaling pathways and also interact with various hormones and growth factors. But in most of the part of the world, especially in the developing countries, pregnant women are deficient in various micronutrients like iron, folic acid, B12, and B6. In our study, we are mainly focusing on the vitamin B12 and folate. Vitamin B12, as we know, cobalamin, it is a water-soluble vitamin mainly synthesized by the microorganisms. And the major sources include animal products like meat, fish, and poultry. And it plays an important role folic acid. The major sources are whole grains, vegetables, fruits, and beans. And it is important for DNA synthesis, cell division and proliferation, placental growth and development, and in the brain growth. Apart from micronutrients, fatty acids also play a very important role in determining pregnancy outcome. And among various fatty acids, long-chain polyunsaturated fatty acids, they have major structural and metabolic functions. Arachidonic acid and docosahexaenoic acid are the major functional LC PUFAs, and these PUFAs are required for the fetal and infant brain growth and retinal development. Fetus mainly depends on the maternal DH and AA supply, as its capacity to synthesize this LC PUFA is very limited. And the fatty acid profile in the fetus is mainly affected by the fatty acids ingested by the mother during pregnancy and lactation. So the maternal intake and metabolism of these fatty acids plays very important role in determining pregnancy outcome. The synthesis, liver is the major site where LC PUFA synthesis takes place. There are actually two series of fatty acids, omega-6 fatty acids and omega-3 fatty acid. Linoleic acid and alpha-linoleic acid are the precursor molecules and are called as essential fatty acids, as they are not synthesized in the body and have to be obtained through the diet. And these fatty acids, like linoleic acid, is converted to arachidonic acid, and alpha-linoleic acid is converted to icosapentaenoic acid and docosahexaenoic acid with various enzymes like desaturases and elongases. They play very important functions in the body. First, the major function is to maintain membrane structure and function and cellular and organelle integrity. They act as modulators of various transcription factors and therefore influence gene expression. And omega-3 fatty acids particularly have anti-inflammatory role and act as pro-resolving and antioxidative components. Now let's see their role in the pregnancy. These LC PUFA, maternal LC PUFA intake and metabolism, it plays important role in determining length of gestation, initiation of labor, and vasculogenesis and angiogenesis. These are major processes involved in the placental development. And from the mother, these fatty acids are transferred to the fetus, and it plays important role in fetal organ development, mainly brain and retina development and overall health. But in case of a low supply or inadequacy of LC PUFA in the mother, it leads to different pregnancy complications, like it may lead to preterm birth, intrauterine growth retardation, gestational diabetes mellitus, and preeclampsia, that is hypertensive disorder during pregnancy. So as we all know, there is a very high consumption of vegetable oil nowadays, which is very rich in omega-6 fatty acids. But around 10,000 years ago, the consumption of omega-3 to omega-6 fatty acids ratio, it was around 1:1 to 1:2. But nowadays, this ratio has been hampered. It is come to 1 to 15:20. So there is a drastic change in the ratio of omega-6 to omega-3. So we really have to cut down this ratio and bring it back to 1:1. So this is the current Indian scenario. India is undergoing a rapid shift in dietary patterns, which is known to fuel the epidemic of NCDs. In India, a high proportion of population is vegetarian due to cultural beliefs, and therefore B12 deficiency, it exists along with low omega-3 fatty acid deficiency. And also there is high intake of saturated and unhealthy fats, which is known to increase the risk for these metabolic diseases. So it is very important to understand the long-term effects of B12 deficiency and omega-3 fatty acids on the cardiometabolic risk factors. A series of studies in our department, human and animal studies, they have demonstrated the link between B12 and omega-3 fatty acids through the 1-carbon metabolism. This is the 1-carbon metabolism. This basically involves the transfer of single carbon unit from folate to various methyl acceptors. In this case, vitamin B12 it acts as a cofactor for the enzyme methionine synthase, which converts methylation of homocysteine to methionine. The methyl groups from methionine are then transferred to S-adenosyl methionine, which is a major methyl group donor. And these methyl groups are then used for the synthesis of various proteins, lipids, and nucleic acids. But in case of vitamin B12 deficiency, the methylation of homocysteine to methionine will not occur, which leads to increased homocysteine level. And increased homocysteine is known to increase the oxidative stress and alter lipid profile. An altered lipid profile is considered as one of the major risk factors for blood pressure and other cardiovascular diseases. Secondly, the methyl groups from adenosyl methionine, that is SAM, are transferred to PEMT enzyme, that is phosphatidyl ethanolamine methyltransferase, which catalyzes methylation of phosphatidyl ethanolamine DHA to phosphatidyl choline DHA. These are the major membrane fatty acid phospholipids. And this conversion is basically important for the transport of L-CPUFA from the liver to circulation and to various tissues. But in case of B12 and DHA deficiency, the methyl groups, instead of going towards PEMT, they are increasingly diverted towards nucleic acids, leading to altered DNA methylation. And as I've already explained, alterations in the DNA methylation is known to alter the level and genes expression of genes involved in various metabolic pathways. So we have, with this background, we have planned an animal study. Actually, it is not possible to carry out longitudinal studies in humans and obtain different tissues from the human samples. And it is also not possible to control the level of nutrients in humans. So we thought animal model is the best model to study the multigenerational effects. And we hypothesized that a sustained long-term B12 deficiency across 3 generations will adversely affect cardiometabolic variables. And this may be ameliorated by a combined supplementation of B12 and omega-3 fatty acids. This is the study design. Actually, we have used a Vistar rat model in this study, and the animals were fed 5 different types of diets. This is the control diet where all the nutrients were given in the optimal amount. This is the vitamin B12 deficient diet, BD diet. Here we have supplemented B12, given B12 supplementation, that is BS diet. And in both these diets, we have added the omega-3 fatty acids, BDO and BSO. And we have used 10 animals in each group. And the pregnant female rats were fed these diets, and they were allowed to deliver normally at day 22 of gestation. Rats usually deliver at day 22 after conception. And after that, they were continued on the same diets. Now, the female offsprings from this generation were again used for mating to obtain F2 and F3 generations. And we have mainly used male offspring for further experiments. This is the diet composition. We have used fish oil capsules, Maxipa as a source of omega-3 fatty acids like DHA and EPA. This is the parameters that we have studied. We have studied pregnancy outcome of the animals, then metabolic variables like glucose, insulin, lipid profile, and blood pressure of the animals, and also studied the liver histology and fatty acid analysis. Now these are the results. We found that short-term vitamin B12 deficiency, it had no effect on the pregnancy outcome. Pregnancy outcome, it mainly includes the weight gain during pregnancy, then how many offspring that female is producing, and what is the weight of that offspring. It is called as litter size and litter weight respectively. So we found that short-term B12 deficiency had no major effect, but long-term B12 deficiency, that means in the F2 and F3 generations, we found that it lowered the weight gain during pregnancy and litter weight. While combined supplementation of both B12 and omega-3 fatty acids, it was found to improve the pregnancy.
Hello.
In high insulin levels. But supplementation with both B12 and omega-3 fatty acids was beneficial. As I have already said, homocysteine is known to increase the oxidative stress. We study the lipid peroxidation marker, malondialdehyde (MDA), and as we can see here, B12 deficiency, it showed significant increase in the oxidative stress, while other supplemented group, they showed improved oxidative stress. Uh, in this case, vitamin B12 deficiency, we found there was increase in the levels of cholesterol, while surprisingly, we found that vitamin B12 supplementation, it increased triglyceride levels. We also studied the blood pressure of these offspring by using non-invasive method, Telcuff method. And as we can see here, vitamin B12 deficiency, it increased the systolic blood pressure, while diastolic blood pressure was similar between the groups. We also carried out fatty acid analysis, and we found that B12 deficiency across 3 generations, it adversely affected the LC-PUFA levels in the liver as well as in the circulation, that is plasma. Then we also studied the expression of genes involved in the lipid metabolism, as lipid, it is the major culprit for most of the cardiovascular diseases. And we carry out gene expression, and we found that B12 led to increased expression of genes which are involved in the fatty acid synthesis. Like these are the genes SCC and FAS, which further increase the levels of saturated fatty acids while decrease the good fats, that is PUFA levels. And it also decreased the expression of genes involved in beta oxidation pathway. So simultaneous increase in the fatty acid synthesis while decrease in the beta oxidation, it led to accumulation of lipid metabolites and therefore causing impaired lipid metabolism. We also studied liver histology, wherein we found accumulation of lipid droplets inside the hepatocytes of vitamin B12 deficient animals, while we found normal liver histology in case of other animals. So to summarize, we can say that vitamin B12 deficiency, it led to adverse pregnancy outcome. It also increased oxidative stress, then homocysteine levels. It lowered the PUFA levels and also altered the lipid profile. Also increased the lipid profile. While B12 and omega-3 fatty acid supplementation, it corrected most of these parameters. So combined supplementation of B12 and omega-3 fatty acid, it was found to improve the cardiometabolic variables. There are also other animal studies which were carried out in our department. This is the preconceptional micronutrient deficient model, wherein B12 and folic acid was not provided in the diet. They were B12 and folate deficient right from the preconception period. And we found that the micronutrient deficiency, it affected the estrous cycle, then ovary and mammary gland development, and it also showed the lower expression of genes involved in placental development. Then we also studied micronutrient imbalanced model. In this, we had a low vitamin B12 and excess folic acid group. And this imbalanced model basically showed increased oxidative stress and altered fatty acid transport. And we also, in this study, we also carried out the neurotrophin studies. We found reduced levels of neurotrophins, then reduced brain DHA levels, and impaired cognition in the offspring. We also studied preeclampsia rat model. This model is pregnancy hypertension during pregnancy. And in this model, we found high oxidative stress levels, then placental inflammation was there, and impaired angiogenesis. This is the process for the placental development. And we also found impaired cognition in the offspring born to the preeclampsia rats. And we also had a high fat model. So all these studies basically showed the importance of micronutrients like folic acid, B12, and fatty acids in the determining pregnancy outcome. So what we need to do now, we already know that nutritional insults or any imbalance in the maternal nutrition during pregnancy, they cause nutritional programming leading to increased risk for the chronic diseases. So now there is a need to carry out a large number of nutritional intervention studies, and it should be implemented all over the world, and as a reprogramming strategies to prevent the adult diseases. And simultaneously there is also need to explore, the research should be carried out to study the nutrient interactions, drug nutrient interactions, how the nutrients play important role in different environmental conditions to improve the pregnancy and offspring outcome. The take-home message is that you are what your mother ate, and good maternal nutrition during pregnancy is important for healthy childhood as well as healthy adulthood. Thank you. Hello.
Hello.
Yes.
Okay. So students, please ask the questions. Hello, students. Students, do you have any questions regarding the topic? You can write in the chat box also.
I think there are no questions. Anandita, has everybody understood?
I think everybody understood very well. So, good afternoon again to all of you. I would like to take this golden opportunity to thank the respected speaker, Dr. Amrita Khaire, for giving this wonderful speech on how nutrients affect the adulthood diseases. Means during pregnancy and pre-pregnancy, a good nutrients eating means taking of good nutrients and good environmental condition is how much necessary for adult life. We understood from this presentation very well. So now I am requesting our HOD to please convey her thoughts, convey her views on the topic.
Ma'am, as you have justified the importance of the first 1,000 days, and also it is correlating with the non-communicable diseases. So, as how you have focused on the maternal nutrition, and how you have emphasized on the other points, like which is related to the community nutrition programs, and also like related to the food and nutrition security, and along with the maternal nutrition, which particularly focuses how the maternal malnutrition is affecting the placental abnormalities. So like overall all the subjects has been correlated with each other, which gave the ultimate benefits of getting the actual desired knowledge to all of our students. So ma'am, thank you so much for spending your busy schedule with us, and your precious time for giving us so much of the emphasized knowledge by yourself. So I really thankful to you, ma'am. So hand over to you, Anita, ma'am.
Okay, thank you, Dr. Amrita. Thank you very much for your wonderful speech, and thank you, students, for your for attending this lecture. Thank you all.
Thank you so much, everyone. Take care. Bye.
Thank you. Thank you so much, ma'am.
So we can leave now. I think we can leave the meeting now.
Okay.